Tēnā koutou katoa. Haere mai rā e te ope o te kāhui ma te pukupuku. Te o tākau whakaihu waka. I runga i te kaupapa o te rānei e, opening karakia. Manakitia tēnei hui, manakitia tēnei rōpū. Manakitia tēnei runanga mō te hauora. Tainai ki tēnei kaupapa, arahina nā pātai, arahina te kore orero, nei kia puta ai te marama, tihei, mauri ora. Basically saying welcome, ask lots of questions around the kaupapa, have lots of discussion, and welcome to the evening. Kia ora.
Thank you, Sandy, for the welcome. My name's Kirsten Capel and I'm with the University of Otago, Christchurch, but based here in Nelson as part of our group of trainee interns, final year medical students who come up to Nelson to finish off their training. And it's with pleasure that we've worked with the Cancer Society colleagues, research colleagues from the University of Christchurch who have travelled up to share some of their knowledge with us today, and with funding from the Care Foundation, this has all been possible. So welcome to you all and those people who are online who have joined us. We're going to do questions at the end. So if you've got a burning question, tap it into your phone, or if you've got a piece of paper, write it down just in case you forget during the course of the lecture. So without further ado, I'd like to welcome Logan, who's going to introduce the research team. So thanks, Logan.
Kia ora koutou. So my name is Logan Walker and I have the privilege of introducing the team here tonight. So the three people that are going to be speaking tonight are members of the Mackenzie Cancer Research Group. We are based at the University of Otago in Christchurch. Here's a photo of our group, and we've got some members here tonight. Our group is a research group. We do cancer research, and our role is to really better understand the disease and try and find ways that can support our community that are having to deal with this challenge. And we do this in many ways, and we interact with researchers in the country, around the world. We interact with scientists and clinicians. We sit right in the medical school, the hospital. We have really close connections with medical friends in the hospital, and it makes for a really productive research environment, which I hope you'll get a better understanding of why I say that tonight. We have a number of students that come through our lab every year, honours students, master students, PhD students. Many will come through, complete their degrees and go on and hopefully lead very successful careers in their own. So that's part of the role that we play. And it's a snapshot in time, that photo. And down below, you can see the number of organisations that support our research in the way of funding. So although we try and get funding through government, you can see the number of organisations, non-government organisations that support our work. And of course, Cancer Society plays a major role in that space. Tonight, you're going to hear research across the cancer continuum. So this is a diagram sort of highlighting the factor that a number of people are cancer-free. We try and understand what sort of factors can keep people healthy. How do we prevent cancer from developing in individuals, particularly those who are at high risk? What about diagnosis? Can we improve diagnosis, improve treatment? What about those who are now living a life post-cancer treatment and thinking about surviving and survivorship? So we've been in this part of town. We were at Motueka last night, and three of us were giving talks, and three talks were given in the grand round at the hospital today, and you're hearing three more tonight. So you can see the names of different researchers that are here and the area of research that they target. So tonight you'll hear from Dr. George Wiggins, Professor Gabby Dax, and Associate Professor Marg Curry. And we hope that you enjoy this evening, and we hope that you really feel free to ask questions and gain as much knowledge as you can from those that are presenting this evening. So I will hand over now to our first speaker, Dr. George Wiggins. Thank you.
Thank you, Logan. Kia ora koutou, ko George Wiggins, toko ingoa, haere mai, and welcome. Just want to start out by thanking everyone that's been involved in organising tonight and all of you for coming out to hear what we do. So I'm a genetic researcher or a geneticist. So to understand what that is, we're obsessed with this molecule called DNA. So DNA is made up of what we call 4 nucleotides or 4 bases, and these are nicely encoded for us as A, T, Cs or Gs. And these are, in each cell we have about 6 billion bases, so 6 billion A, T, Cs and Gs, and these are made up of 3 billion pairs, so you have 2 copies of every base. And the DNA itself is kind of the guide or the instruction manual into how that cell will behave, and so how that living being becomes. Between any 2 individuals, any 2 humans, we have about 1 in 1,000 bases that differ. And it's this variation that captures the diversity that we see in this room or we see in any population. These variations can also account for some of the inherent risk that we have for diseases. And this includes breast and ovarian cancer, which is where I work. But not all variation is the same. And so I've taken this excerpt from the Nelson Regional Development Agency, Nelson Tasman, the understated jewel of New Zealand, where those in the know of where to go. Renowned for its stunning landscapes, sun-soaked weather and convenient accessibility, it provides a serene escape away from the crowds. So a nice little excerpt and advertising slogan from the Nelson region, although today's weather might be an exception. So we could add some sort of variation to this sentence and it will change the way we read it. So you could have a simple typo. So this is where the sun in the sky has changed to sun as in your child. But all of us looking at this paragraph would pick that up as a typo, and we understand that the context of this paragraph hasn't really changed. We could have other simple typos that might make this a little bit more sinister or a little bit more worrisome. So we could change sun to gun. Maybe that would turn some people away from coming to Nelson, might encourage others to come. But we can also have larger variation. And this is where we can delete large segments of the DNA or the paragraph. And this can completely change the context of this paragraph. So now instead of being an undercrowded region, this is now stating that Nelson is the jewel of the crowds. So the opposite of what the Nelson Regional, whatever we're trying to advertise. Sorry, I forgot the name. But as I was saying, all of this comes back to the work in breast and ovarian cancer. And today I'll focus mainly on breast cancer. And so this is an infographic from the Breast Cancer Foundation New Zealand. It's just giving some facts and figures, and it's nice to understand how common this disease is. So this is the most common cancer in women. This is figures from 2022. It says 9 women diagnosed daily. That number's already gone up to about 10 women diagnosed daily. So this is still rising. But what this figure doesn't capture is that a big portion, or about 5 to 10% of these breast cancers are due to heritability, so something that has been inherited from either their mother or their father. So as a cancer geneticist, if I knew the DNA or that guide sequence to every person in the room, we could start stratifying the population and start understanding the level of risk that each individual would have for the disease. And we could start grouping those people into people that would have a less risk than the general population, those that have the same level as the general population, those that have a slight increase, and then we can identify those that have really, really large increases. And then we can, knowing this information, we could start to tailor the clinical need and the clinical resources towards the relative risk of these people. So we could reduce screening for people that were likely at lower risk, or increase screening, or for the people at high risk, we can offer really drastic prophylactic surgeries. And these are quite drastic things. The research that I'm focused on is actually working within this high risk population group. And so women that carry variations in these genes, BRCA1 and BRCA2, can have a really, really large increase in developing breast cancer. So we're talking 70 to 80% lifetime risk of developing that cancer. So throughout their lifetime, 7 out of 10 women will develop breast cancer. But there is this little cohort of women, about 30%, that will never develop disease. And it's why we're interested in why these 30% aren't developing disease. So what about these women is protecting them from that high risk variation? And when we can understand what that is, can we leverage that to offer preventative therapies to that whole cohort of everyone that's at high risk? To do this research, we have to work as a global team. The sheer numbers that we need is we would have to identify every single person in New Zealand to even come close to the cohort size that we can get from these international consortiums. So we're involved in this consortia called SIMBA. It's about 55 study sites throughout the world. I've got pins on the world there for each of the study sites, and you can see the little red pin down in Christchurch, which is the only study site in New Zealand. And all of these research groups are joining forces to try and understand how we can modify the risk in these high-risk people. So now we've got this group of high-risk individuals only. So we've got these people with these variations that are causing big increases in risk. And then we look at the rest of their DNA to try and understand, are there other variations in that DNA that are either enriched for disease or enriched for not developing disease? And what we're focused on with those large, what we call copy number variants, so like deleting those words out of that paragraph before. What we found in this study was that we found a deletion over a gene called SILT1A1. The name of the gene doesn't matter, but its function is actually involved in estrogen metabolism. And we know that the breast tissue is an estrogen responsive tissue. And so the logic here is quite nice, and it seems to be making a nice story. So what we're trying to test now is whether we can treat, at the moment in cells, with 2 copies of the gene to see if they can mimic the same response. So can we inhibit the SILT1A1 protein to mimic a deletion and see if we can get that protective response? How we go about doing this, we have multiple sort of strategies in which we can attack this effort. And we've got really simple cell-based models in the lab. So we can have what we call cell lines, and these grow in little petri dishes, sort of stuff like you see in the movies. We have cell lines that have that really high-risk pathogenic variant, and then we have the general population level risk cells. We can treat them with a compound that's meant to inhibit that SILT1A1 gene, and then we can measure a bunch of cancer phenotypes to see if we can capture that protective effect. The problem with these methods is they don't translate great into what's going to happen in a person. And so we've got to develop more sophisticated methods. And so this is where the 3D model, so that second sort of panel, comes into play. And so this is where women that are undergoing mastectomies in the Christchurch hospital can donate tissue. So they're consented to donate tissue. And we take a small bit of normal tissue, so surplus to clinical need, and we can actually grow that into a 3D structure using scaffolds. And so it kind of looks like a mini breast in the flask or in the petri dish. And then we can do similar sort of treatments with drugs. And so we'll be collecting tissue from women that have these high-risk mutations, as well as the ones at general population risk. And what we have glossed over is that from those big studies, there's more than just SILT1A1 that was interesting. So we've got hundreds of genes that were showing protective or increased risk effects. And to go through these one by one is really, really time-consuming and laborious. And so instead of doing that, we can develop these high throughput methods where we can actually throw 70,000 gene inhibitors at a pool of cells, and then we can use really complicated statistical methods at the end to decide which ones look promising and which ones don't. The ultimate goal of all of this research is going back to that general population, and we want to push those women that have really high risk mutations back towards a general population level risk. We want to try and avoid the need for those prophylactic surgeries. A lot of people don't uptake them. They're quite invasive and life-altering. And at the very least, if we can delay these until after childbearing years, that would be the bare minimum that we would want. I'd like to, just thank you all again for coming here. This is a team effort. So this is the Mackenzie Cancer Research Group this year. A bunch of funding bodies there. Thank you.
Kia ora, kia ora. Thank you everybody for coming. Ko Gabi Dax, toko ingoa. Guten Abend, herzlich willkommen. Goedenavond. So I'll be talking to you about my favorite molecule, which is vitamin C. So the overview tonight will be on, does it have any role to play in vitamin C? And we have evidence for and against it. It's not as easy to sum up as all that. So I'm very much aware that vitamin C is one single micronutrient that we need to stay healthy, and that there is a lot of other nutrients, but I'll only be talking about vitamin C tonight. So we're one of the very few species that are unable to make vitamin C. So your dog, your cat, your mouse, your guinea, your rabbit, they can all make their own vitamin C, but we have a mutation that prevents us from making vitamin C from glucose, which is what all the other species are able to do. We and the great apes, guinea pigs, and zebrafish. So we need it on a daily basis, and there's a range of fruit and vegetables that are really good at providing us with vitamin C. For example, the ones on the left, so the yellow peppers are very good, the kiwi fruit's very good, the oranges. Then intermediate would be like broccoli and kumara, etc. Unfortunately, as you go further to the right and you go towards my favorite fruit and veg, the chocolate, it's got no vitamin C whatsoever. So on a daily basis, we should aim to grab some stuff, several of the ones on the left. So how much do we actually need? Well, the Department of Health here has recommended a minimum intake of 45 milligrams of vitamin C a day. Now that is very little, and it will prevent scurvy, but it is not a healthy level. And the rest of the world are at 90 milligrams or above, so about double of what we have the recommended daily intake for. And then there's the in between, inadequate still. Even if you take the 90 milligrams a day, in general, that's not quite enough. So in order to get saturating levels in your blood, you should aim for a kiwi fruit and say a pepper each day. That would do. That's enough. So what does that translate into? So we measure vitamin C in the blood of people, so in plasma levels. So less than 23, it's called micromolar, that measurement of vitamin C. You're starting to move towards being scorbutic. 11 micromolar, you will definitely see signs of scurvy. If you go for the saturating level right at the bottom here, that's the level that the Ministry of Health actually recommends for people that have chronic diseases. So that can be achieved through diet. So we are looking at what does vitamin C actually do. So most people are aware that it is an antioxidant, so it defends you against free radical damage. Collagen production is a very important function of vitamin C, and that is where the obvious signs of scurvy come to fruition. So if you have bleeding gums from scurvy, that is because the collagen is not formed properly. It's also important for immune function and neurotransmitters. Those functions, vitamin C carries out as what's called a cofactor for enzymes. Now, an enzyme is a catalyst, so it does things in the cells. So in order for your cells to be healthy, those catalysts, a lot of them require vitamin C for functioning. So we've gone out to measure vitamin C. So a lot of studies are done by pure surveying of people's dietary habits. We believe that's not quite good enough. We actually need to measure what's there. So this first one here, we looked in blood, so in plasma levels, and we had healthy controls, a reasonable size. And then we had 3 groups of patients that have cancer. And we tested them either before their surgery for their cancer, or during chemotherapy, or during immunotherapy. Now, this patient group down here are patients with advanced cancer, in this case, melanoma. Then we could categorize the vitamin C that we could measure in that plasma into what's enough, or if it's less than 23 micromolar, as I said, that's not enough. And healthy people have about 11% of the population do not have high enough vitamin C in their blood. And that is similar to cancer patients before surgery. However, once patients receive chemotherapy, so their bodies are under stress from both the chemotherapy as well as their cancers, 16% of that population have low levels of vitamin C. And the patients that are in advanced disease and receiving quite a severe type of treatment, almost a quarter of them have these very low levels of vitamin C. So for clinicians, this is an important thing to keep in mind when they have a patient in front of them, to make sure that that is not a case that adds to the burden of the cancer. We've done a lot of work with He Taonga Tapu, the Cancer Society Tissue Bank in Christchurch. That's a wonderful resource that we have access to, following ethical approval and approval from the Tissue Bank Board. They collect samples from patients undergoing surgery in Christchurch Hospital, and virtually every patient gets the opportunity to donate part of their tumor for research for unspecified future use. So we have looked at brain cancer, breast cancer, kidney cancer, bowel cancer, and uterine cancer, and measured in frozen samples of these cancers the amount of vitamin C in it. We also looked at markers of cancer regression, so we can look at certain proteins. In that respect, we can, in many cases, compare the tumor and the adjacent normal tissue to see whether there's a difference. We can show an association between what we measured, so the aggressive phenotype as well as the vitamin C, to what else we know about the tumor, so the grade, the stage, the size, the vascularity, etc. And we can also look, because these patients often have donated their samples many years ago, we can do a follow-up and see how well these patients have survived. So this is how it's done as an overview. So during surgery, tumor is removed. In this case, this is a colorectal cancer. We only need a tiny little sample for our research. So it's the size of a pinprick that we can do all our research with. In this case, it gets ground to a fine powder, it gets put into a lysis buffer, and then we can run it through this machine called an HPLC, high performance liquid chromatography, and that very, very accurately can measure the vitamin C in that tissue. And we get these sort of graphs to measure it with. So this is what we found out. This is an example of the brain cancer research that we've done. So these are primary brain cancers. Looking at these graphs down here, so every single dot is a separate patient sample. And on the y-axis, we've got the amount of vitamin C that we measured. And down below here, we've grouped them into either the lower grade tumors, so the less aggressive ones, and the higher grade tumors, the more aggressive ones. And in this case, it's the glioblastomas. And we found that the vitamin C levels in the glioblastomas were significantly lower than in the lower grade tumors. We also were able to measure, we call it the hypoxic pathway, it's one of the aggression markers. And we found that if there was more vitamin C in the tumor, that aggression marker was on average lower. So there was an association with more vitamin C reducing, dampening that hypoxic response. So as an overview of all the other studies we've done, we could show that the vitamin C levels varied greatly between patients. As you could see from the previous one with the dots, there was quite a spread. So we often work with the average or the median. We found in brain, bowel, and uterus that lower vitamin C was associated with higher grade in these tumors. So the more aggressive type of tumors had less vitamin C in them. And there was a link also between low vitamin C and some of the prognostic factors. So the grade, the stage, the size of the tumor, for example. We also saw that there was an association with overall survival. So in this case, I'm showing brain cancer survival. So the way to read these graphs is, this is the probability of survival. So they all start at 100% probability. And then over time, each time this step goes down, the patient has deceased. And what you can see here is that the patients with higher vitamin C, so the green top line here, survive a little bit longer than the ones with the lower vitamin C, which is the red line. We've seen something similar for breast cancer. But again, you can see that these graphs are not that far apart, but there is a trend, and it's significant, it's statistically significant, that they tended to live longer if they had more vitamin C in the tumor. That's important. This is where we could measure in the cancer itself, in the tissue, which is where we think it acts as this cofactor for the catalysts that need to function healthily. However, we've also looked in the advanced cancer patients with melanoma. And in this case, we don't have access to the tumor tissue. We only looked in the plasma. And in this case, we see no difference at all between the survival of the ones that had more or less vitamin C. We don't believe that's necessarily got to do with the melanoma as a disease itself, but it has to do with the fact that we measured it in the blood and not in the tissues. Now, it's very important to point out association is not the same as causation. And this here is a very good way of showing that. So the bottom here says chocolate consumption per person in the country. And the y-axis here is the number of Nobel Prizes won by people in that country. It's the most beautiful association here, very strong. So the more chocolate you eat, the more likely you are to get a Nobel Prize. So it's clearly not the case. We need to look at causation. So in order to do that, we need to actually change the amount of vitamin C that goes into a tumor. So we need to kind of nudge the bear, really. So in the first instance, nobody's ever done that. We need to look at how much vitamin C actually reaches the tumor when a patient is provided with a high dose infusion, which is what a lot of alternative practitioners provide. We then need to look at whether that actually changes any of these aggression markers that we are interested in. And finally, and this is outside the remit so far, we have not been able to do that, does it change the survival outcome of these patients? So there's an important difference between the amount of vitamin C you can achieve by orally ingesting, so by diet or by supplements, which is in the bottom here. So your body is very, very good at capping the amount of vitamin C that goes around in the plasma. It secretes it very effectively through the kidney. However, if you give these bolus doses of very high vitamin C via infusion, we can get 200-fold increase in the plasma concentration of vitamin C. But you can also see, this is a time scale here, that it goes back down to base levels within 24 hours. So is this peak enough to make the tumor have more vitamin C? And that was our main question. For that, we did a clinical trial in patients with colorectal or bowel cancer. And the study protocol meant we got a small sample during the colonoscopy that we could test for the amount of vitamin C in it. Then patients were randomized to either receive the high-dose vitamin C or not, the control patients. And then for 4 days immediately prior to their planned surgery, they got high-dose vitamin C. In this case, it was 1 gram per kilogram. So that meant most of these patients got 75 grams of vitamin C daily. It's a huge, huge amount. And then during surgery, we could get the tumor. And in this case, we could also sample right across the tumor from the edge of the tumor towards the center in order to see whether there was any variation in vitamin C levels. So in order to make it more understandable, I've color-coded this. So green, the top is the high vitamin C that we measured. So these are accurate measurements, but I'm just showing it by color versus the lowest. So these are the patients that are control patients that, so you can see here, this is the tumor, and that was sort of at mid to low range, and the surrounding mucosa was in a similar range of vitamin C. And unsurprisingly, because there was nothing done with these patients, they had similar levels of low vitamin C across. Importantly, the center of the tumor, which is poorly vascularized, had very low vitamin C, very low vitamin C. Now, in the infusion cohort, we started off with very similar levels before the infusion started, but after the 4 days, we managed to get vitamin C right across the normal tissue as well as the tumor. But again, we see this range of vitamin C levels as it goes towards the center of the tumor. And that's probably important to realize, that it doesn't reach all parts of the tumor as well. So the take-home message for today is that many healthy adults as well as patients with cancer do not consume enough vitamin C and thereby have low levels of vitamin C in their blood. There is an association of increased levels of vitamin C in tumor tissue with a less aggressive type of cancer. We can increase the amount of vitamin C in tumors by infusion, although again, we saw quite a range between patients, so not everybody will benefit as much as the next person, and we are investigating why. So we still do not know whether high vitamin C levels will improve patient survival from cancer. And this is our team that has been working on the vitamin C story, and our funders on the right. Thank you very much.
Oops. Okay. Okay. Well. Okay. Kia ora koutou, ko Margaret Curry tōku ingoa. I'm just trying to think what I'm going to do with that, but I think if I stay still, it'll be fine. My usual interest in research is the tumour microenvironment, and that's all the cells within the tumour that are supporting and helping the cancer cells to grow and spread. But what I'm going to talk to you about tonight is a completely new area of research for me, and that is whether we can prescribe exercise to improve cancer treatments. So recently, with the improvements in early detection and diagnosis and treatment, we've got a lot more people now who are surviving with and beyond cancer. And that's brought up a couple of health concerns. The first is, can we reduce the toxicity of cancer treatments so that patients don't have to have the short and long-term adverse side effects of their cancer treatment? And the second is, can we improve the efficacy of the few treatments that we really do have available to us so that the patients don't suffer the recurrent disease and progressive disease? And so in the last 20 years, there's been a lot of research gone into exercise, and it seems like exercise may be a very useful adjuvant or additional therapy in cancer management. And this has led to a new field called exercise oncology. So it seems really counterintuitive, doesn't it, to exercise while you're having your cancer therapy. And now we've got very strong evidence that shows that exercise, or regular moderate intensity aerobic exercise and resistance exercise during and after cancer treatment, not only preserves your cardiorespiratory fitness, but it also decreases a lot of the cancer-related adverse effects like cancer-related fatigue, depression and anxiety. It increases physical function and strength, and therefore improves overall the quality of life. And this evidence is now so strong that the American Society of Clinical Oncologists have gathered together an international panel of experts, and they have stated that oncology providers should recommend that patients do aerobic and resistance exercise during treatment to reduce the effects of side effects.
There's also emerging evidence starting to come out that shows that exercise following diagnosis may improve treatment outcomes or treatment efficacy. And this comes from dozens of studies in over 68,000 people with cancer. And it's shown a very strong, consistent trend of a reduction in risk of cancer recurrence, reduced risk in cancer-specific mortality, and reduced risk in all-cause mortality. But the trouble is, none of those studies were actually designed to look at that particular question of can exercise increase the efficacy of treatments. So we still have to be a bit cautious about this, and we can't say for sure that that's the case. So more research is needed in that area. So the current recommendations for people with cancer for exercise are actually pretty much the same as for anybody in the general population. Avoid inactivity. Progress towards regular physical activity with the aim of reaching and maintaining at least 150 minutes of moderate intensity exercise a week. And that sounds like a lot, but it's actually less than 30 minutes a day of just brisk walking where you can chat to your friend as you're going. Also recommend 2 to 3 moderate to vigorous intensity resistance exercise sessions just to build up muscle strength. But the important difference here with cancer patients is that exercise really needs to be tailored to the individual. And you can imagine that because sometimes people are 45 and they have cancer and sometimes they're 85. And so the exercise required will be quite different. The other thing that they recommend too is that it's an accredited exercise physiologist or a specialist physiotherapy that actually prescribes and delivers the exercise, so they can just keep an eye on people and make sure that they're safe. So this is Jess Allen, who trained as an exercise physiologist in Australia, and she worked there for 6 years before she came back to New Zealand, and she enrolled to do a PhD at the University of Canterbury with Professor Nick Draper and Dr. Kate Reid, and myself at the University of Otago in Christchurch. And she was funded for that PhD by the Cancer Society. And most of the research that's been done so far about exercise and cancer has been done overseas. So what we really wanted to do with Jess is see if it was feasible to prescribe a personalised exercise program to people during treatment for early stage breast and colon cancer, and most importantly, within the clinical setting here in New Zealand. So if you look at that blue arrow from left to right and imagine that as the sort of journey that somebody goes on through their cancer treatment, that first they're diagnosed with cancer, and then there's about 10 to 14 days, possibly slightly longer now, before their first treatment, which in our case was generally surgery followed by about 24 weeks of chemotherapy. And during that 24-week period, Jess designed a 12-week exercise program which had aerobic and resistance exercises in it and some mobility and balance too. And the patients could then pick whether they'd do 1 to 3 sessions a week, and most did 2 to 3. With the orange arrows along the top there, those are the physical assessments that Jess did for each patient as they came through. So first a baseline assessment for when they came in and were diagnosed. Then there was one just after surgery to make sure that people were fit enough to go into the exercise program. And then every 4 to 6 weeks after that. And from those physical assessments, Jess then set the exercise program for that individual so that they should have a training effect as they went through their chemotherapy. In addition to those assessments, we nicked something from High Performance Sport New Zealand, and this is something that a lot of the major sports teams do. Every time they go into the gym, they have a very quick survey that just said, did you sleep well last night? Did you eat well last night? How was the last exercise session? And according to those answers, Jess just tweaked the exercise program for the individual depending on their mood of the day or feeling of the day. So was the exercise program feasible? It was certainly challenging because it was right during COVID-19 pandemic. So Jess had to deal with lockdowns and social distancing. And of course, the patients are really scared of getting COVID-19 while they were on treatment in case that delayed them. But in the end, Jess invited 65 people onto the program, mostly women, and that was because there was one lymphedema specialist that really believed in the program and she kept referring people through. 39 accepted and 29 were eligible according to the inclusion criteria that we had for health. So only 23 people completed the program, the 12-week exercise program, which doesn't sound like a lot, does it? But when you think that they were doing 2 to 3 sessions every week for 12 weeks, that was over 500 supervised 1-hour sessions that Jess was taking. So it's a lot of work for her. We had really good recruitment and completion rates, which compared really well with international stats. And the adherence rates were really good too. So patients picked how many times they were coming in a week, and they pretty much did that, except if they had unplanned surgery or they were really ill after a round of chemo. So we had no serious adverse events, which confirms what international studies have shown, that so long as it's a specialist that's giving this exercise program to cancer patients, it's very safe. We showed improved flexibility, especially in the shoulder joints for women who'd had breast cancer surgery and treatment. There was some improved well-being and energy, but most importantly, we improved the physical function from baseline. And as you can imagine, most people, when they come into surgery and treatment, they have a huge drop in their physical fitness that they then have to work at to bring that back up. And sometimes they never achieve the same level that they had before they went in. And with this, we managed to keep nearly everybody at least at baseline or slightly better as they went through their cancer treatment. So the patients who went through, and I admit it was slightly a biased selection, but they loved it. And there were 3 themes that I've just put up there. The first is very common. It gave me some control over what was happening to me at a time I didn't feel I had much control over anything. The second is interesting to me in that a lot of patients mentioned that they just didn't know how their body worked anymore after surgery and treatment. So they really appreciated having that one-on-one session with the exercise physiologist to teach them how to exercise safely again and to get them back into it, or to get it, actually start exercising, because some people had never exercised before. The other thing that was really interesting is it didn't matter how much we told them that it was a really good idea to do this program. It wasn't until their treating specialist or their doctor said it was a good idea, and then they thought, aha, okay, we'll do that. So the recommendations that came out of Jess's PhD were, of course, that it had to be a very individualised and person-centred program. As I mentioned, clinical advice was really important, and the patients didn't want to come back into hospital for their exercise sessions. So we had a beautiful gym in the new section of the hospital looking out over Hagley Park. But everybody said, we've been in hospital for treatment, we don't want to come back in for exercise. So thinking about that 23 people with 500 hours of supervision, we decided that the best way to do this was to start with one-on-one, maybe 6 sessions where the exercise physiologist would go over the exercises and teach them to the patient. And then they could go into small supervised classes of maybe 4 or 8 people that still had an exercise physiologist looking after them all, and then into the open gym, still under some supervision. And the other thing that we realised is really important is the social side of it. And everybody really appreciates having a group of people that are going through similar sort of experiences, I suppose. So while we were doing Jess's study, we met an awful lot of people in Canterbury who are all really keen on this idea of having an exercise program for people who are being treated for cancer. So we've set up a collaboration called the Cancer Prehabilitation and Rehabilitation Aotearoa, which is a terrible mouthful, and we had months of fighting over what we were going to call it, but it really describes what we're trying to do, and that prehabilitation prepares people for the treatment, rehabilitation allows recovery. And in that, well, who's involved? Lots of people from the hospital. We had oncology, surgery, cancer care nurses. The University of Otago in Christchurch and the University of Canterbury have been involved with lots of people from different departments. Active health physiotherapy. the Southern Cancer Society, Te Papa Hora, our health precinct in Christchurch, and also actually the Christchurch City Council has really been behind this too, which has been great. And in that sort of floret of photos of people that are involved, right in the middle is Professor Bridget Robinson, who's a medical oncologist who's just retired, and she has really been driving this for many years. So the aims of our CPRA collaborative are to develop high quality evidence-based exercise programs for everybody as they go through all sorts of different cancers at different stages. And to do that, we really need to grow our research program so we can define the prescription that's necessary for each person with exercise as they go through their treatment. We also want to develop opportunities for students to come in and do clinical placements if they're med students or physiotherapy students, and postgraduate research projects too. And all this will help to build a very small exercise oncology workforce. So ultimately, what I really hope is that we can have a multimodal cancer prehabilitation, rehabilitation project, care bundle, I guess, which we wrap around patients as they go into their treatment. So they get nutrition advice, an exercise program, and psychological counselling. So we've been running the collaborative for a year, and there are a number of opportunities and challenges, and I've listed 3 of the challenges there. The first is funding, which is always our problem. We've managed to get the logo, as you can see up on the top left, and a web page, which we're going to have situated with the website of Te Papa Hora, which is our health precinct. I've managed to get 0.5 salary for 2 years for our exercise physiologist, Jess, to keep running the exercise programs. And we've also got some money from a pharmaceutical company, AstraZeneca, to run a similar feasibility study for men with prostate cancer who are undergoing androgen deprivation therapy and radiation therapy. And I would just add that's the first time I've ever accepted pharmaceutical money, but yeah. Venues and gym spaces, we've been really lucky. We've been supported well by Te Papa Hora and the Christchurch City Council, and I think the Christchurch City Council will see that if they can show that their swimming pools and gym spaces are being used for community health, it's a really good thing to promote. And on the middle photo there, you can see our new sports facility that's being built in Christchurch, which is called Parakiore. And it's the second biggest in the Southern Hemisphere after Melbourne, and that's going to have gyms and also exercise labs. So we'll be able to run the program there. And it's only about 5 minutes walk from the hospital, and it has plenty of parking. So it'll be a great venue to have when it finally is built. And the third challenge is really the availability of expertise. And we don't have a lot of exercise physiologists who are trained in New Zealand. So Professor Nick Draper at University of Canterbury is setting up a first in South Island clinical exercise physiology training course. And we're really hoping that that will start in the second semester of next year. So as always, there's just heaps of people to thank. There's a big collaborative, and in particular, I've highlighted Jess Allen, who's our exercise physiologist. She's done all the work that I've been talking about today, and our funding bodies, and of course, the people who participated in the study. Thank you. Thank you. Okay.
What a fabulous series of talks. I've learnt a lot, and I hope you have all learnt a lot. And just a little bit of plug for Nelson. I saw in one of the newspapers recently that Nelson has got the most active population, but there's a big but. It's still not good enough. So I think only about 30% of us in the Nelson region, 30% of our population have reached recommended levels. So we've got a long way to go. So we're good compared to the rest of the country. How bad's the rest, Christchurch? So hopefully you've got a number of questions there. And so how are we going to run this? If you could just let us know. We've got people online who'll be asking questions as well. And then our speakers will come up to the mic and share their expertise. So welcome, Gabby, George and Marg. So do we have a question? There we go. We've got some mics.
Maybe start with them.
There we go.
Hang on, we need to get this.
Sorry, can you speak? We'll need to have the mic because so then the people online can hear you.
Hello? Yeah. I'm just asking about, you mentioned the fruits to give you the high percentage of vitamin C. What about the tablets? When you go into the pharmacist and you buy the tablets, are they effective as the fruits are?
Yes, very much so. So the regarding vitamin C in isolation, the tablets will give you the exact amount that it says on the tablet. They're highly bioavailable, they're highly water soluble, they work. However, your fruit and veg give you a whole range of other micronutrients. They give you fiber, they give you all the other goodness that would be missed out with the tablets. So if you are unable to eat the healthy fruit and veg, by all means, go for the tablets.
Right. Thank you.
We've got, how many mics have we got? Yep, no, I can.
Yes, so my question is, it showed clearly that the infusion of vitamin C doesn't reach to the nucleus of the tumour. From my understanding, I saw the shading and in the nucleus it was. So is it possible to inject the tumour directly with vitamin C?
I think you probably can, but not all tumors are accessible, and it's not, I mean, it's sort of slightly simplified in that picture because the areas that are low in vitamin C are not necessarily in the center only. I mean, in these tumors, we found that there was a gradient from the outside to the inside. But we also know that the areas that are low in vitamin C in a tumor are the same ones that are low in oxygen. Now, with oxygen, we know it's away from the nearest blood vessel. And so it's the same thing for vitamin C. As it travels through the tissue, it gets taken up all the cells and it doesn't reach the far ones. And those areas can be spread around the tumor. So it's not as easy as that. What we've done is 4 days of infusion. You could possibly increase that. And it was also a one-off 4 days. So if people were to consider this, and if we ever find that it is actually doing what we think it should be doing, you would go for longer periods.
I understand. Thank you.
I'm going to go to a question online now. And we've got another vitamin C question. So the question is, can healthy bodies consume too much vitamin C?
I would say healthy bodies, no, but patients that have advanced cancer, people that have, like burns victims, for example, were tested. And even though they were having a good intake of vitamin C, some of them took supplements, their plasma vitamin C levels were still borderline scorbutic. So if your body is fighting a disease, is fighting infection, is fighting some sort of stresses, they will consume a lot of vitamin C. There's anecdotal evidence. Now, I don't know whether this is true, but animals that can make their own vitamin C, for example, a goat. There's a story saying a stressed goat can make up to 70 grams of vitamin C a day. So animals that can produce their own vitamin C, they will up the production in order to battle that stress. And yeah, I mean, throughout your body, different types of cells require or take up a lot of vitamin C, whereas other ones less. So for example, the white blood cells, they accumulate a lot of vitamin C and they're required for their function. Your brain accumulates a lot of vitamin C, it requires it for the function. Whereas other parts, for example, your muscle is lower in vitamin C in general.
Another question here in the audience. Thank you.
A non-vitamin C one. I'm just curious in terms of the exercise benefits. I realize that the New Zealand sample is very low, but from the international samples, non-vitamin C, sorry. Non-vitamin C. In terms of the exercise, did you see any benefit variation in terms of the patient's age? For example, do specific ages get better benefits from the exercise regime, and or did it depend on the baseline? Did healthier, more fit people come out better in the long run or get better benefits, or did those who weren't exercising actually have better benefits from the treatment or during the treatment with the exercise?
With our particular study, we didn't take it far enough to see that really, because we were just trying to see if it was feasible to do the exercise while they were going through their clinical treatment. But in international studies, certainly they have shown that definitely, no matter the age, the stage, whatever, so long as you're exercising after diagnosis, then you have reduced adverse effects with treatment, and there's quite a lot of evidence, but as I say, not so strong for the survival outcomes. So, yeah, does that answer your question? Thank you. I don't want to go, I'm up here now.
I could ask you a question.
Okay.
Tēnā koutou katoa. This is, you don't, it's for you, Professor Curry, so you don't have to sit down again. With enough, there seems to be you presented enough business case to warrant a center of your dream wraparound support. So what do you think is the barrier that for any government funding for it to continue? And I think all of you can all combine your researches here and do the wraparound support in that one super center. So what do you think is the barrier for that?
There's one major barrier, and that's nobody's
Kia ora.
going to make money out of it. So the oh gosh, the drug companies won't make money out of any of that. So that makes it very hard to get funding. And so what we really probably need to do is, as somebody raised today during the hospital talks we gave, we need to get a health economist to come in and show that actually this is saving the government money by stopping people having to come back through for more treatment. And that's quite a challenge, but that's probably what we need to do next. So one thing I think we either need government funding, which is probably highly unlikely, or maybe in Australia, insurance pays for this sort of thing. So if we could persuade the insurance companies to start giving some money towards exercise programs or other vitamin C or whatever, that would be really helpful. But I think so long as we're in one centre and not national, it'll be very hard to persuade the insurance companies that that's a good thing for New Zealand. Yeah. Thank you.
Yes, I think, yeah, it's complicated, isn't it, these things? And I think the wider support for these good programs, and it's not just about the bottom line dollar, is it? It's about quality of life, and that's where our economists can help. So, yes. So I'll just go to another question online here. Just bear with me. Sorry, I thought I had another one, but I don't. So we'll take the lady up the back there in the,
I've got a mic.
oh, sorry.
Yep. Just another
Sorry,
question
get
about
on.
exercise. How important do you think it is using an exercise physiologist, which really limits the uptake? I know in Nelson, the Cancer Society use a physiotherapist. And I guess, you know, it's not just cancer patients that would benefit from exercise, but everyone, but particularly like heart failure patients and post-MI. And so, I mean, potentially even you could be using like a health coach situated in general practice.
Yes, I think you're right.
Or a hip.
It probably depends what stage of cancer there is. And I think with later stage cancers where there are a lot of other health issues, then it's really important to have somebody like an exercise physiologist that understands the biology of what's happening. But I think we can probably start training personal trainers and so on especially for the early stage cancers. And in fact, during COVID-19, we were thinking of doing telehealth and doing videos and things like that. So there are certainly ways that we can explore that. But at the moment, we're trying to keep it as safe as possible because the last thing we need is an adverse event to sort of destroy all the work that we've been doing so far. Yeah, I agree.
And so I think our lady up the top there.
Thank you. Thank you for your participation and bringing this to us today, this evening. I'd just like to ask a question around exercise. I know that from a cardiac point of view, getting patients to come along to exercise programs because they'd already been out of work. And for the cancer patients, then they're out of work for a lot longer because often with the chemotherapy and the surgery. So how do they get along to these programs when there is funding to get them to the classes?
Yeah, that's a major problem. And there are places like Pink and Steel where you can apply for grant funding to get money to go to the programs. And I think for people like me, it's easy because I can afford to do these things, but there are definite barriers to a lot of people to getting along to an exercise. I mean, they may be working mums who have young kids, so they're trying to work, look after their family, and they simply don't have time to come along to extra classes. So yes, that is a big problem. And that's really what I think needs to happen. This needs to be something that's free and freely available to everybody. So I'd like to see it as part of the standard therapy for cancer. Thank you.
I think we've got okay, up top and then down.
Just a question for George. Can you just talk us through the availability or how, you know, for the BRCA gene testing, and would it be appropriate to just sort of do a grand sweep of genetic screening for people? How do people access that test? And
so clinically, you'll get a genetic test based on the age of your cancer, your family history, and either your GP or your treating oncologist will be the one that would refer you for genetic testing. I think it's probably our feeling or thought that everyone that has breast cancer should be getting genetic testing. So Logan had done a study, he spoke about it in Motueka, but we miss about half of the people that have a BRCA1 or BRCA2 variants that have breast cancer. So we're missing a lot, and that affects families. And so the more we do, the better.
I think we had a question down the front here.
Hi, just a comment about exercise class availability in Nelson. We do have funded exercise classes through Pink and Steel for both men and women. We're currently running 2 a week, and we changed the time of the class slightly from term to term to try and capture the particular group that are enrolled, so we can try and get as many people in to a time that suits them. So some of our classes are early morning, some of our classes are evening, We have had groups at lunchtime, and sometimes that suits people with young children who, they might have to collect from school in the afternoons. But I think the biggest challenge, as always, is funding. But I would really encourage anybody who wants to know a little bit more about it to either contact myself at Activate Physio or to go onto the Pink and Steel website, because there is availability for funded support in Nelson.
Yes, I certainly don't want to say anything negative about Pink and Steel, because it's a fantastic program. And but my point really is I feel that it should be freely available to everybody, and it should be part of our health system, and that's, aiming for the highest that we can, I think.
Yeah, and absolutely.
Thank
I
you.
mean,
Good.
we've just produced through a Physio New Zealand advocacy document looking at equity and looking at the disparity across the different regions, with a suggestion to ministry that this needs to be mainstream medical treatment.
Oh, fantastic. Well done.
It's just a matter of packaging it up to present to Whāmek, isn't it? It's not a packaged pill, but it is a package of care. I've got another couple of questions online here. So for Gabby, and this person is interested if you've done any research regarding vitamin C and radiation therapy treatment, specifically as doctors have mixed opinions on patients having vitamin C during treatment due to the antioxidant properties.
That's a very good question, and that's exactly what we're planning on researching next year. We've got an honours students to look into that. There are hypothetical reasons why that could be a bad thing, but the timing of ascorbate administration and the radiation, I think, is what's vitally important to understand. So what we'll be doing in our research for next year is we're just going to, again, measure the amount of vitamin C that patients have in their bloods just before and just after radiation. Because if radiation does interact with the ascorbate, with the vitamin C, we would expect a drop in what we see in the plasma levels. So we'll be actually attacking that by measuring what's really going on. A lot of the data that people refer to is data done in mice. Now, mice, we all know, can make their own vitamin C, so their picture is quite a lot more complicated with regards to a lot of vitamin C work, but specifically with the radiation and vitamin C work.
Thanks. And another question online is, and I think it's for you again, Gabby, does a Myers infusion of vitamin C help for cancer treatment?
I am not familiar with Myers infusion, I'm afraid, but the robustness of clinical data regarding whether high-dose vitamin C infusion is beneficial is still not that good. There are individual case studies and small clinical trials where benefit has been shown, both for improved outcomes, so longer survival, but mainly a stronger study in my eyes was that it reduced the side effects from chemotherapy for patients with endometrial cancer. But there's very few good studies out there, and unfortunately quite a few poorly designed studies that compare to historical data, which is not the correct way of doing it.
Any more? Question here.
Thank you, George. I was interested to learn more about the SIMBA study, and I'm curious to know how you imagine the learnings from that study, how long it will take to actually inform improved cancer prevention, treatment care, and so on.
Yeah, the age-old question of timeline. Yeah, we're hoping to push things as fast as we can towards clinic. So the research that we're doing at the moment is taking that direct result from the SIM study, trying to test it in the lab. If we get promising results, then we can start looking at doing sort of phase 1 clinical trials. And then it will be sort of maybe 5-10 years. So it's a slow process. Yeah, Mark just mentioned. Because these are preventative therapies, we don't want to be giving toxic chemicals or toxic drugs. So we've been focusing on using either repurposed chemicals that are used commonly. So for Salt 1A1, I didn't mention, one of the inhibitors for that is a component of the oral contraceptive. And so women are already taking this quite regularly. So we know it's well tolerated, as well as some food compounds, so turmeric and a few others can inhibit it. So it's just about whether we can deliver it to the site.
I guess mine was a similar line with Simba, George, and AI. Do you think the AI can it advance your genetics and your testing faster? You know, you say like, I was asking for the timeline, but AI is developing so fast. And because you're worldwide and you have so many, you know, you said it takes a lot of hours and time and resource, but could AI speed that up at all, do you think?
Potentially. We're not there yet, so we need to have well curated data sets to train AI, and we don't have that at the moment. It's because we're looking at the whole genome, so there's 6 billion bases that we're looking at. A lot of those regions of the DNA are poorly understood. We do use some AI tools to help try and understand some variants already, but even those are showing quite a large amount of error. And so, yeah, once we get better curated data sets and we understand things a lot better, then AI can start to be trained and then things will speed up.
We have a question up the back there. And those of you online, I encourage you to answer questions too, ask questions.
Hi, another vitamin C one. I just wondered what your thoughts are on vitamin C infusions already being used in practice. I guess is that ahead of evidence?
Unfortunately, I mean, it is administered in the community by a lot of alternative practitioners. Very few of them measure anything. At best, the plasma levels are measured and nobody's ever measured whether it reaches the tumor. I have not seen evidence of survival benefit. So it is, the great advantage of vitamin C is it is safe. It's safe for anybody who has working kidney function. If your kidney is not working well, you have to be a lot more careful. But people have infused, I mean, we've gone for 75 grams per day, and the worst side effects we had was a slightly raised blood pressure due to the amount of liquid and salt being infused. So it is a very safe, and it has been shown worldwide that it's a very safe treatment. So besides the financial damage that you can do, it is a good, it's not a toxic way of going about it. But as I said, the robustness of the clinical evidence is pretty poor at this point, and it needs to be done well. People need to do good clinical trials rather than just treating everybody who wants to be treated and then asking how they've felt. Because most people that would have undertaken the vitamin C infusion will say they felt really good, but it's nothing objectively measured.
So we've got another question online here. I think this one's, someone's just saying that there's informal dance fitness for women, gold coin donation per session, or whatever you can miss towards power, costs every Monday and Thursday at the Haven SDA Church side entrance in Grove Street. So anyone who's interested in that.
Just comment to that actually, because it's really interesting. We went out to the Pacific community and asked about exercise, and they had a provided exercise group, but what they really wanted was a dance group. And it's a great way to get exercise. So dance groups are it.
And also that social benefit that you talked about.
Exactly.
Another question here. Someone online here is currently taking exercise sessions in the Selwyn area in Canterbury under the IHS. Our ladies are loving the session and are certainly feeling and seeing the benefits during the treatment, after treatment, and also end of life. The group is feeling supported and love the cuppa catch up afterwards. All the benefits you mentioned, Prof. Mark. We are currently running this session for FOC with Cancer Society funding, the venue hire. I would love to undertake more sessions as I find this so rewarding. However, funding would also be helpful. So that's someone who's
That's a lovely comment.
providing fantastic comments. So thank you, Hamish, for providing that comment. So I think, how do we convince our government that this is important? Any more questions here? I think we've got one up here.
Just about the exercise, maybe a suggestion would be to start with the funding. I know that when I was getting the radiation in Christchurch, I went to the Christchurch City Council swimming pool and they provided a free week of membership, and that was a way of getting in the water and cooling down after the radiation session. So that could be just a quick option. I know it was something that wasn't suggested, it was something I thought about to try and cool things down as it was an intense process.
Thank you. That's, yeah, a great comment.
And I think it was pleasing that Mark did acknowledge the Christchurch City Council. And as I say, I mentioned Nelson City Council because they were talking about wanting to support. So it's not just the health sector, it's also our wider community. So it's good to get that buy-in. Another question. Here we go.
Sorry, I just thought for the last 7 years, the Cancer Society Nelson Tasman have been, we sort of realised quite early on that exercising during your treatment was really beneficial. That we'd read some overseas studies. And so we provide yoga 3 times a week in our office, free for anybody going through treatment. And we would like them to go while they're in treatment, but also after treatment. And we also offer an 8-week gym program as well for anybody that's going in treatment. If we, while we're having treatment, we suggest that, but also afterwards, if that's to help them with their rehabilitation, as you say, especially after breast cancer, or, to get the mobility back again. So we offer that free of charge in our community, as well as in Motueka and Golden Bay.
Oh, that's fantastic. And Kirsten did say that Nelson was the best for exercise. So there you go.
Any more questions? Oh. Yeah, is it? Yep. Okay. Without further ado, before we have our close, I would like to thank George and Gabby and Marg for travelling up here to Nelson. I've learnt a lot tonight, and I think from the questions and discussion, we've all learnt a lot tonight, and will, I think, potentially motivate us to try and do something here. And I know there has been whisper that this team is quite interested in even setting up research up here. So I put another challenge to the team. So if you'd like to join me in thanking our group before we close formally. And there's a cup of tea afterwards.
I'll just do a wee closing karakia. Kia whiakaria te tapu, kia wātea ai te ara, kia tūru ki whakataha ai, haumi e hui e, pai ki e.
Thank you to those online, and sorry we can't share a cup of tea with you, but I encourage you to go and have a cup of tea at home. And so we're offering a cup of tea out in the foyer, so you're welcome to stay and ask the team more questions. And thank you very much for coming up this evening on Guy Fawkes Night. Mhm.